today we're going to have a look at one of the key organ systems. That's the cardiovascular system. So in this session, we're going to have a look at the constituent components of the cardiovascular system. We're going to have a look at cardiac anatomy, and we're going to look at some basic cardiac physiology to include the cardiac cycle, cardiac output and its control. We're also going to have a look at the anatomy and physiology of the vascular system, and we're going to end up talking about the physiology of blood pressure. So this diagram is showing us the different components of the cardiovascular system. And the two key components are the heart and the blood vessels. Cardio pertaining to the heart and vascular pertaining to vessels. And this is a general overview diagram of which we will investigate in more detail as we go through. But first of all, I want to talk about the cardiovascular system as a whole, and the concept that the cardiovascular system consists of two separate circuits. So blood is pumped around the body by the heart, but the heart pumps the body in two loops. There is one loop that goes from the heart, from the right side of the heart here, to the lungs, and back again. And the other loop goes from the heart to the rest of the body. So we have like a pulmonary loop to the lungs, and we have the systemic loop to the body. Okay, now is the time to have a look at where the heart is. Now the heart sits substernally. You can probably see just about here where I'm drawing the notch at the bottom of your breastbone. You can probably feel that in your chest now. And your heart is substernal to that underneath. It's pointing off towards the left, and with the apex downwards towards the left. Okay. A lot of people tend to think the heart's sort of over here somewhere. That's where they tend to put their hands when they say, "Oh, I've got a bad heart." But in fact, it's central. Okay, it's in a central cavity. It's in a central cavity between the two lungs. This diagram is just showing you some of the key features. And I think that the key features that you could take away from this diagram is that, first of all, the heart is surrounded by its own membrane, the pericardium. We will look at that a bit later. That it's attached to a whole mass of large blood vessels at the top. Okay, and those are both arteries and veins. Okay, and you will also notice shown here that the heart has its own special blood supply called the coronary circulation, and here are some coronary vessels that I'm drawing here. So this is the human heart. If you want to have some idea of scale, a really good measure for a healthy human heart is that it's about the same size as its owner's clenched fist. So if you've got a healthy heart, clench your fist, your heart is probably about that size. One of the problems we have with people misunderstanding the size of hearts is that when they look on television, such as Holby City or Casualty or whatever, and they see a heart, it's usually quite big. But that's because it's a diseased heart that they're working on, and diseased hearts are usually bigger hearts. The only other hearts that are bigger are those of really, really fit athletes, and those are bigger because the muscle's more defined. So we're going to start a journey now into the heart. And the first thing that we need to describe are the membranes that surround it and protect it. And the protective membrane of the heart, in fact a series of membranes, is called the pericardium. Peri meaning around, cardium meaning the heart. So the pericardium has two layers. An outer layer, which is largely tough connective tissue, it's known as the fibrous pericardium, and then inside a double layer with fluid in between called a serous pericardium. Now, we've met a serous membrane before, because when we looked at the meninges, of the central nervous system, they have a similar configuration. Remember, they had a tough outer layer and thinner inner layers. So this is a repeating pattern in our anatomy, that we have coverings, protective coverings, but also inner fluid containing membranes too. Now, the reason for having this fluid, and you can see it here on this diagram, having pericardial fluid, is it allows the heart to move inside this protective membrane. It allows the heart to move inside the body with some quite dramatic movement without causing irritation or abrasion. Okay, so if you didn't have this fluid-filled membrane, your heart would literally wear away at the membrane, and because of all the friction caused by its beating. So these protective membranes with fluid in them are very important. There is a condition that affects the pericardium, which is actually quite painful, and it's called pericarditis. Oh, can't do me T's. Pericarditis. So pericarditis, very painful disease. Sometimes it can be mistaken for a heart complaint, and in fact it's a bacterial or viral infection of this membrane that becomes inflamed, and the movement of the heart causes the pain. Okay, so that's our outer covering. Now, the majority of our heart is made up of muscle, and that muscle layer is called the myocardium. So the whole thickness of the wall of the heart is cardiac muscle. Now, on this diagram, you can see I've put one actual slide of real cardiac muscle that's been stained, a microscope slide, that's here, and a line drawing here that describes what's going on there. So let's have a look at the line drawing. First of all, we can see the individual myocytes. Myo, if you remember from our anatomy lecture, muscle site cells. And to give them their full name, they're the cardiomyocytes. So they're the cardiac muscle cells. And you can see their nuclei, they're indicated here. You can see striations, which is the repeating pattern of actin and myosin that's present, needed for contraction. But you can also see how they're all woven together at different junctions. And these junctions are called intercalated discs. And they're marked in blue on here. But hopefully you can see, and I'll draw them in on the actual diagram, here, 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 etc. You can see those lines all over this picture. That's where the individual cardiomyocytes are joined together. And you can also see here the nuclei. So you can also see the nuclei of the cells. And you probably can make out, I'll draw a line around it. If you look in here, these striations, you can see these stripes. And that's characteristic of cardiac muscle cell. So the whole wall of the heart is made up of that. And it's the contraction of this that causes the heart to beat. Okay. Now this diagram shows a heart that's been cut open. And the reason I've chosen this diagram is because it shows you the inner layer of the heart, the inner membrane called the endocardium, inner part of the heart, within the heart. Now the other thing you should notice is the thick wall of the heart. That's what we've just been looking at. So that's the thick muscle layer we've just been talking about. And here you can see this rather milky coloured membrane, smooth, glassy-like appearance. In fact, this is abnormal. In reality, you wouldn't see this milky colour, but this is a thickened endocardium. So this is probably an example of, guess what? endocarditis, the inflammation of the endocardium. Normally, it would be completely transparent and all you would see would be a glossy covering. The purpose of having this smooth covering on the inside of the heart is to allow the blood to flow smoothly around the heart and not cause any irritation or turbulence. Because you just have to remember that blood is not a liquid, it's a liquid containing cells, and those cells moving at speed can bump into each other and bump into the walls of the heart and blood vessels, and repeatedly over time that could cause irritation and damage. So the endocarditis is the inner membrane, again protective and helping the cardiac muscle to work as a functional pump. Okay, the next diagram is some of the key anatomical features of the heart. Now, what I'm going to do is I'm going to describe them, but I just want you to realize just a few of them. Okay? So first of all, we need to know that there are 4 chambers to the heart. Okay? So 4 chambers to the heart. 1, 2, 3, and 4. And these ones over here are wrapped around the other side, so you can't see them. But they're effectively 4 chambers of the heart. So at the top, RA and LA stand for the right atrium and the left atrium. Okay, now if you've ever been into the Portland Square building here in Plymouth, you'll notice that there are 3 big areas outside the lecture theatres that we call the atria. And it basically means a collecting space. So you find an atrium in a building, and these are collecting chambers. This is where the blood first enters the heart from the 2 circulations. So down below those are the right ventricle and the left ventricle. Okay, so the atria are there to collect the blood, the ventricles are there to pump it. Okay, what you'll also notice is that there are a couple of blood vessels here. Okay, so the blood vessels are connected to those different chambers. So here, VC, SVC, and IVC. S stands for superior, I stands for inferior, and we saw that in our anatomy, or you should remember from your anatomy, that superior means above and inferior means below. What does VC stand for? It stands for vena cava. I like to think of it as the venous caves. So the cave, the cavernous veins. So effectively they're the biggest veins in the body, some of the biggest veins in the body, and blood is being collected from the body and going into the right atrium from here. What happens then is that the blood passes to the right ventricle and then out something called, we've got PT, which literally stands for the pulmonary trunk. Pulmonary standing for the lungs. And up here, what we do is the pulmonary trunk splits into the left and the right pulmonary artery. Okay, so these are, oh, I can't even spell, pulmonary artery. So that's a P. P-U. Pulmonary artery. So that those vessels are now taking blood away to the lungs to have them have the blood oxygenated. The blood, once oxygenated, will then come back via the 2 pulmonary veins. That's a better P. pulmonary vein. Okay, so here we've learned another bit of key anatomy from the body. Any vessel taking blood away from the heart we call an artery. Any blood vessel coming back to the heart we call a vein. Okay, so a very simple set of definitions. So these vessels, the pulmonary veins, then come here into the left atrium, down to the left ventricle, and the left ventricle, very, very powerful muscular pump, pumps things up via the aorta, the biggest artery in the body. A or T. Okay. So those are the key things I want you to talk about the heart. The four chambers, atria and ventricles, right and left. The veins going into the heart from the body, from the lungs, and the arteries going away from the heart, the pulmonary arteries, and then the aortic, the aorta. Okay. Okay. So I sort of hinted at what happens here in terms of blood flow, but this diagram shows it really quite neatly. And the arrows show you where that blood comes from. So first of all, blood comes into the heart from the rest of the body. So this is blood that will have lost a lot of its oxygen and is packed full of carbon dioxide because it's transferred its oxygen and picked up carbon dioxide from the rest of the body. And it will enter into, first of all, the right atrium. Okay, so our blood collects here, and then when there's enough of it to knock open this valve here, it pushes down into the right ventricle. Okay, then when the heart contracts again, off it goes to the lungs. Okay, so in the lungs it gets lots and lots of oxygen, and then the blood returns back to the heart to the other side. So basically it returns to the other side, then down into the ventricle, and then when the heart contracts again, off it goes to the aorta. So a single blood cell travels from the vena cava to the right atrium, to the left ventricle, to the lungs, to the left atrium, to the left ventricle, and then off into the aorta. So that is the sequence of blood flow through the heart. Now this doesn't work, blood doesn't flow in the right direction without some very, very important tissues. And those are here, and those are the valves. And we'll see them on the next slide. Okay, so this is a view of the top of the heart. The vessels have been chopped away. Okay, so the vessels have actually been chopped away, and what you're looking at is you're looking down on the top, and you can probably see LV here, left ventricle and right ventricle. Okay, now what I want you to notice is two different types of valve here. Now these two valves here, 1 and 2, are known as the atrioventricular valves, because surprise, surprise, they're between the atria and the ventricles. So these are the atrioventricular valves. They're there to stop blood from being pumped back out of the heart, so therefore allow blood only to enter the heart through into the atria. Okay, you can see that the types of valves here, these two valves here, the mitral, or what we call bicuspid valve, bicuspid because it's got two components to it, 1 or 2, and this is the tricuspid because it's got 1, 2, 3 cusps. Okay, those are valves that allow blood to go into the heart but not back out, so that the blood is being pushed in one direction. Now when the heart contracts, it pumps its blood out via these two valves. From the right ventricle here, if you remember, the right ventricle off towards the lung, if you remember, and these are what we call the semilunar valves. They're called semilunar because perhaps you can see that there's a slight crescent moon shape. And in fact, when you look at them, if you were to look at them sort of across ways, they look like this. They look like little cups. And what they do is they allow blood to come out of the heart, but the little cup prevents blood coming back because the blood collects in the cup and closes the valve. So you can see that here. You can see this little indentation. So that's where blood would get caught and close the valve. So we've got a non-return valve. So blood goes out of these but can't come back. So the combination of the two valves allow blood into the heart in one place and out of the blood in the other place, and prevent it from returning where it came from. So the blood always goes around in one direction. Okay. Two little things to notice here. This is just the first part of the aorta here. This is the biggest artery in the body. See these holes here? You can see these holes. These are called the coronary ostia, and they're in fact the entrances to the coronary vessels. You can see here, one and here. So the coronary circulation. So this is the arterial portion of the coronary circulation, which takes freshly oxygenated blood to supply the whole heart muscle and to keep it healthy. Okay. Now the other thing I want you to notice are the heart sounds. Why now? Because it's the closing of these valves that causes those sounds. When the heart contracts, it squeezes the blood under pressure. Now what that means is that the first heart sound is caused when these valves here, 1 and 2, slam shut. It's rather like closing the door. Once you close the door, you get against clunk, you get that sound. Now the other heart sound that you get is the closure of the other valves, the semilunar valves. So the two heart sounds, the lubbed up that you hear when you listen to somebody's heartbeat, is closing of the valves under pressure. So it's the closing of these heart valves under pressure, causing turbulence and a sound that can be heard through the chest. Okay, come on to something now called the cardiac cycle. It's important to understand, and I hope the diagram shows you, that the heart doesn't contract independently. So the two sides of the heart contract at the same time. So basically, this happens in coordination. So the first bit is atrial systole. Okay. Now a systole is the contraction. So this is first of all slight contraction of the upper part of the heart, which is forcing blood into the ventricles. See, the white arrows show you that the blood is now collecting in the ventricles. Okay. The next thing you get is you then get a slight contraction of the ventricles, followed by a strong contraction of the ventricles. So it's like a pre-contraction and then a really hard contraction. And as a follow of that, which is called ventricular systole, that's when blood leaves the heart. Okay, so blood's leaving the heart to the lungs, and to the rest of the body. And this word, systole, because this is the main contraction, is the origin of the word systolic. So your systolic blood pressure, the top figure, the higher figure, is a pressure caused by this contraction. So squeezing the blood out of the body, out of the aorta under pressure into the arteries. So your arterial blood pressure has a systolic pressure. And then the next thing that happens is the heart relaxes and the ventriculars relax and allow for blood to follow, to fall through in what is called ventricular diastole. And surprise, surprise, this relaxation causes a drop in pressure, and this is what causes the diastolic pressure reading. So the heart is not contracting, so the blood is not under as much pressure, so you get a diastolic pressure. So your diastolic blood pressure is a measure of the general pressure in the system, not a function of how hard the heart is squeezing. Okay, and that in some ways is the reason why that lower figure in the blood pressure is more important, because it tells you the general pressure of the system overall. The heart is always going to exert a high pressure when it in systole, so the higher figure is not necessarily important unless accompanied by a higher diastolic pressure. Okay, so that's the cardiac cycle. Now, how does it work? How do we get a state where the heart can contract and relax, contract and relax, contract and relax, contract and relax? How's that all put together? Well, it's controlled electrically. Okay. Now, if we look at the heart, we can find areas of the heart that are more electrically active than others. And there's one particular part of the heart that is more excitable and more active in terms of contraction than anything else. And it's called an area called the SA node. And SA stands for sinoatrial node. SA node, sinoatrial node. Other thing this region is known as is the pacemaker. So cool, because it sets the pace of contraction for the whole heart. It does this by sending electrical signals across the heart in bundles of specialized muscle cells which conduct the electricity, okay? And they cover the top of the heart. So you can see the first part of that is the electrical conduction across the top of the heart, which causes the atrial contraction. So it causes the muscle on the top part of the heart to contract. There's another region here called the AV node, AV standing for atrioventricular, if you remember. We talked about the atrioventricular valves. So because it's between the atria and the ventricles. Now when the impulse gets to that, a very dramatic electrical event happens that spreads the electrical impulse all the way through the muscle of the lower heart. And it does it through special bundles of fibers. Okay? And there's a left bundle and a right bundle. And some of the conditions, heart conditions that you can pick up on ECG are things like bundle blocks, where you don't get the correct passage of electrical activity. Okay? So this is how the heart controls itself, by a rhythmic series of pulses of electricity that make the heart contract. Now, the beauty of having this electrical conduction system is that the electricity, the electrical impulse, diffuses through the body's tissues, because the body is made of water. So the body conducts electricity, So we can actually attach measuring devices in different parts of the body and measure this electrical activity. Now we call that the electrocardiogram, or ECG. Now this is just a diagram to show you where we place the electrodes. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and you can have 12 or 15 lead ECG, and remembering that the lead doesn't mean a wire, it means the reading of electrical activity at different parts of the body. So that's what a lead means in terms of an ECG. Okay, so you can see here we've put electrodes in different parts. So those electrodes actually measure the electrical conduction of the heart at different times, and they can measure different levels of contraction. We can see that on the next diagram. Okay, so let's basically just look at this bit down here first, down the bottom. Let's not look at the electrical waves at all. Let's just see the events that we've talked about. So the first thing, if you remember, was the first event is that wave of electrical activity that goes across the top of the heart. Okay. The second thing that we'd noticed was that wave of activity reaches the AV node, and then you get that very strong passage of contraction through the bottom of the heart. And the bottom of the heart is very muscular, and there's a lot of electrical activity coming down the bundles here in the middle. And then the heart relaxes. So the heart then tends to electrically go back to normal. See, so there's not much happening there. There's a slight electrical activity. So when we look at the ECG, we can actually see those events. So the P wave here is the first part of the ECG, this little deflection caused by the atrial events. Then there's this very dramatic QRS complex in the middle, okay, which is caused by this rapid electrical change due to the that accompanies the contraction. And then finally we get this T wave, which is an electrical recovery event. Okay, so it's showing you that things are going back to normal. So we get, when you look at a normal heartbeat, chains of this different type of activity. 3, 4, 5, 6. An ECG can tell us a number of things. First of all, it can tell us the heart rate, so how many beats there are per minute, for instance. So you can see here 1, 2, 3, 4, 5, 6. 1, 2. I can't draw twos. 3, 4, 5, 6, 7, 8, 9. So there's more beats in the same period in this ECG. And this one, 1, 2, 3, 4. So this is a faster heartbeat than normal. This is a slower heartbeat than normal. But we can also determine whether or not it's a regular heartbeat. So here we can see there's a beat here and a beat here and a space, and then the heart going back beat, 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 beat. So we can actually measure not only the speed but the characteristics of the heartbeat. The next slide shows you some of those abnormalities. Okay, so here's some abnormal ECGs. We can use the EKG. Americans tend to use EKG quite a lot. So this is tachycardia. So this is basically a very fast heart rate. So you can see lots of peaks, boom, 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 boom. A tachycardia is generally a heart rate over 100 beats per minute. Now, one of the other things that you'll know about heartbeats is that when you watch a lot of medical dramas, when somebody has a trauma to their heart, or their heart is unwell, they can often go into fibrillation or ventricular fibrillation, and that's when they tend to put on the paddles and pass an electrical current through in what's called defibrillation. So this is what fibrillation looks like. So here are a couple of normal heartbeats. Then all of a sudden, the heart is unable then to continue a coordinated heartbeat. So all of the heart muscle starts to independently contract. So you get this sort of wobbling effect. Now what happens is because it's not coordinated, blood is not passing around the body effectively. So a person, when this is happening to them, is unable to maintain a reasonable circulation, unable to maintain blood flow to important parts of the body. Now the other thing that we can get is a heart block, and I talked about that earlier. This is often due to a previous heart attack or some sort of disease or an infection that affects the heart muscle. And it affects the heart muscle so that the message, or the electrical message, doesn't fully get to the ventricles to allow for contraction. So if you can see, what we've done here is, here's a normal one, P, QRS, T. And then you can see P, nothing, P, nothing, P, QRS, P, nothing, P, QRS, P, nothing, P, QRS. So you can see here and here, we're failing to get ventricular contraction. So we're failing to get that QRS complex there, we're failing to get it. And that's a heart block. It's characteristic of a block in the conduction of the electrical signal across the heart. Okay. So that's our introduction to sort of cardiac function in a way. So we know what the heart looks like. We know how blood passes through it. We know that it passes through via valves and it goes into different blood vessels, and the whole of that pumping is coordinated through the cardiac cycle. And the cardiac cycle is the result of electrical activity causing muscle contraction. And we also now know that you can measure that electrical activity and determine the operation of the heart and its electrical conduction properties. Now the next slide gives you a very brief list of the most important things that determine normal heart function, and we'll have a look at those. So here is your minimal list for a normal heart function. Okay, the first one is the contractions of the individual myocytes must occur at regular intervals and be synchronized. And I think we've already seen that you need a regular contraction of the heart to provide blood for the body, and it needs to be synchronized. So you can't have fibrillation. You can't have this unsynchronized contraction of myocytes because you don't pump suitable amounts of blood from that type of unsynchronized contraction. The valves must be fully open, because if the valves aren't open, you can't actually get the blood through. So blood doesn't move effectively through the heart, and therefore blood doesn't go out of the heart in enough quantity. 3, the valves must not leak. Because the valves prevent blood from backtracking, from going back where it came from, if they leak, what happens is blood doesn't progress. It doesn't progress through the heart on its normal journey. So if they leak, then blood leaks through. It causes turbulence. It also reduces the efficiency of the pumping of the blood, because the individual chambers are not sealed off, and therefore the blood's journey is hindered. Now 4, the ventricular contractions must be forceful. So in very diseased hearts, they contract, but they're not strong. They're not strong contractions. They're not strong enough to push the blood around the body. So they've got to be forceful enough to push blood around the body. And finally, the ventricles must fill adequately during diastole. So there must be enough space in there, and there must be open enough to fill with enough blood to pump on the next stage of the journey. And again, in very diseased hearts, sometimes the walls of the heart become so thick that there isn't much space in the ventricles for blood. So as the walls become thicker, less blood is able to be pumped out every time the heart beats. So these factors 1, 2, 3, 4 and 5 are the key factors that determine normal heart function. So anything that affects 1, 2, 3, 4 and 5 on this slide is going to cause abnormal heart function. And things like heart attacks, things like bacterial infections, things about damage caused by, say, virus infections, are all contributory factors to making normal heart function less likely, because it affects one or more of these different factors. Okay, now we've looked at cardiac function, and we've looked at the factors that determine normal function. We do need to talk a little bit about cardiac physiology. So we do need to talk about what is it that actually the heart does in terms for the physiology of the body. Well, there's a very important physiological parameter, and it's called cardiac output. Now, cardiac output is how much blood is pumped out of the heart over a certain period of time, usually a minute. So how much blood is pumped out over a minute period? Because cardiac output is important, because cardiac output should match the demands of the body. Say, for instance, you're doing very, very vigorous exercise. The muscles in your body and the tissues in your body are using a lot of energy. So they're going to need lots of oxygen, they're going to need more blood, more nutrients, and they need to get rid of their waste products. So they need a higher cardiac output. Okay? When you're at rest, you need to lower your cardiac output because the demand is not so much. Now, there are two key determinants of cardiac output, and these are the ways in which the body controls the heart function. The two key components are heart rate and stroke volume. So cardiac output equals heart rate times stroke volume. Okay? So heart rate is the number of beats the heart makes in a minute, and stroke volume is the number of milliliters of blood that goes out of the heart every minute. Okay? Sorry, every beat. So if you've got 70 beats per minute, so roughly average heart rate, and that every beat is 70 milliliters, 70 times 70 equals 4,900. Okay, I think so. Anyhow, so 4,900 mils per minute. Okay, so with 70 beats per minute and 70 mils, the cardiac output is 4,900 milliliters per minute. So that's the cardiac output. But if you imagine, if you change that, if that becomes 60, then this figure then starts to become 4,200. Okay. But again, then if heart rate goes up 80, this figure now then becomes back up again to 48. So you can see the two are interdependent. And this is the way in which the body matches its blood needs to the contraction of the heart by changing heart rate and or stroke volume. So in extreme cases, the body will increase both heart rate and stroke volume to make sure that there's enough blood pumping around the body. What you need to know now is, how does it do it? How do we change cardiac output? So first of all, the first way in which we can control these parameters of heart rate and stroke volume is via the nervous system. Okay, and the key part of the nervous system we've already seen this week, the sympathetic nervous system. And if you remember, the sympathetic nervous system is that helps the body, prepares it for action. So it prepares it for action in terms of fright, fight, or flight. But basically, it is a protective mechanism. Okay? So the sympathetic nervous system is heavily involved with controlling the heart. The next diagram shows you that quite dramatically. Here we go. So here is the heart, and what you can see is that it's heavily influenced by the sympathetic nerves. Okay, and the sympathetic nerves attach and control the heart in a number of different places. Okay, so the sympathetic nerves can actually increase or decrease heart rate directly. Okay, and then the other part of the nervous system, the parasympathetic nerves, particularly the vagus nerve, which is cranial nerve 10, okay, remember we saw the cranial nerves earlier, they actually control this heart rate by, if you like, tuning it down. So the heart has a large number of nerve endings on it, and the two parts of the autonomic nervous system, the sympathetic and parasympathetic system, work together to provide the control of the heart rate and stroke volume. So that's a key way in which we control how much blood is pumped around the body via the nervous system. The other way we can control the heart or influence the heart is chemically. So there's a number of chemical substances that influence the heart rate. So first of all, adrenaline. We've probably all seen emergency medical dramas where they inject adrenaline to try and get the heart to work properly. But we also know that if we have a rush of adrenaline in our body, if we've had a fright or something, or we get excited, then our heart races, because adrenaline is a potent increaser of heart rate. The other thing is the hormones that are produced from the thyroid hormones. And an increase in thyroid hormone can actually raise your heart rate as well. And there are some important ions, calcium and potassium. So calcium and potassium ions also have a powerful effect on the heart. I think, if I'm right in thinking, one of the medical shows, I'm a bit of an addict. I think it was either Casualty or Holby City, which was last week. Somebody wrongly put up a potassium infusion. Now, potassium has a significant effect on the heart, and it affects the electrical activity because it's an ion. You see, it's got a positive charge here. So it affects the electrical activity in the cardiac muscle and can have a severe effect on the heart if there's too much of it around. Okay. So that more or less completes our look at the first part of the cardiovascular system. That is the heart and heart physiology. Now we need to go to the vascular system. This is a really favourite diagram of mine because it's a picture taken through a microscope of the various capillaries in the body, and it's a very detailed picture of blood capillaries, and it shows you how extensive the network of blood capillaries is, and it also shows some very important features of the capillary circulation. But we might come back to that a bit later on when I can explain the significance of this diagram. So let's of all now look at the vascular system, the blood vessels of the body. Okay, so this shows us in a number of different ways the different types of blood vessels in the body. So first of all, it tells us that we have arteries and we have veins. And as I said before, arteries take blood away from the heart, veins take it back. And we'll see that they are tubes with muscle around them, okay? But one of the things that you will always note, and you can see it in this microscope picture, this is an electron microscope picture, that where you have a vein and an artery together in the same place in the body, you can always tell which is which, because the artery has a smaller, what we call luminal diameter. You see that? This is much bigger in the vein, and a thicker wall. Okay, so arteries, smaller lumen, thicker wall. This is just showing you how that's made up of different tissue layers. But the key things that you need to know is that arteries and veins are joined up via some other blood vessels. Blood leaving the heart goes through arteries, which then branch down to microscopic vessels called arterioles. And then from the arterioles, it goes into what we call the capillary bed. So these are the very fine blood vessels I was talking about just a minute ago, the capillaries. And this is where all the physiological activities take place in the capillary bed. This is where all the nutrients are exchanged, where all the gases are exchanged, where any antibodies move in and out. This is the business end of human physiology, the events that go across the capillary bed. And once that's happened, blood then drains back via microscopic veins called venules, and then back to the heart. Okay, so there we go. So from the heart, through arteries, arterioles and capillaries, back to venules and veins, and back to the heart again. So it is a circulation. Let's have a look at those two types of vessels, the two biggest types of vessels in two pictures. Okay, so you don't need to learn all of these names. It's just to show you the different arteries of the body and to know that they are given names, and some of them you will probably need to know more than others. Okay, so for instance, you might need to know about the femoral artery to take a femoral pulse, for instance, femoral artery here in the groin. You might need to understand things about the coronary artery, because we talked about that. Coronary artery disease is a significant disease. But we might also need to understand some of the other arteries of the body. So for instance, aortic aneurysms often happen in this region here. So that's where the artery balloons and can possibly burst, which is life-threatening. Anyhow, here's a basic diagram, and you should just notice that there are two types of arteries really in the body, elastic arteries and muscular arteries. The elastic arteries are there to really take the force of the blood as it comes out of the heart. They need to be elastic because the heart is pumping, squeezing blood at high pressure, and they need to resist that. The muscular arteries are there to provide motive power to squeeze blood through and help squeeze blood through in other areas. And they're muscular so they can be controlled. So actually it's possible to control the amount of blood passing through them via nervous or chemical signals. Okay, now the next slide is the veins. And what you've probably immediately noticed is there's a great deal of similarity between the two pictures. One's just coloured blue and one's coloured red. And that's true. The blood vessels tend to follow each other and be seen together, as we saw in the diagram a while ago. Again, a number of different veins here, which you'll learn about as you go through your course. Okay. Now, veins are different from arteries in one important other way, and that is that veins, being responsible for the return of blood under a lower pressure than arteries, they need mechanisms to stop blood flowing backwards because of little pressure or sometimes little pressure to help push the blood forwards. So what they need is valves. Okay. Now this shows you a normal vein with its valves. So blood can go upwards towards the heart through the valve. However, it can't return backwards, because as it pushes backwards, it closes the valve. Now, for a number of different reasons, maybe because of genetics, maybe because of other lifestyle factors, sometimes veins become dilated and their valves become leaky. So blood tries to get through and doesn't always succeed. And therefore, these have pools of blood, larger pools of blood than normal in them, and this is the cause of what we term varicose veins. And you can see quite dramatically here how the veins have become distended, and the blood is pooled into these veins. So this is pretty dramatic. But it is just to show you that veins have a set of valves to enable the flow of blood to go in one direction. Put this diagram in because I think it explains some important relationships and dynamics of the vascular system. It's a diagram that shows you a diagram of the blood circulation from the artery, or through the arterioles, through the capillaries, through the venules, and back through the veins. But it also shows you underneath a series of readings there. So for instance, it shows you the blood pressure across that set of tissues. So blood pressure in the arteries, as you can see, remains relatively high until it gets to the arterioles, where there are many, many more of them, and therefore it spreads out and the pressure drops. And it drops even more, and it drops even more, drops even more, through the capillaries and out there. Okay? The other thing that's useful is the velocity of the blood, how fast it's going. So you can see the blood, once it's left the heart, starts to slow down, but only small amounts until it gets down to the arterioles and capillaries, and then it slows right down. And the reason why it slows right down is so that all the exchanges can take place. So there's enough time in the capillaries for things to move in and out of the blood. And then it picks up again when it goes back into the venous circuit. The brown line here is showing you another thing altogether. That's showing you the surface area of those vessels. Okay, so the surface area of the large arteries is quite small. Similarly with the small arteries, when we get towards the arterioles, we see this dramatic increase in surface area as the arteries split into many, many hundreds and thousands of arterioles and then millions of capillaries. So the amount of surface area here available for exchange is huge. So there's a huge surface area of blood vessels to enable physiological events to take place, for gases to go in and out, nutrients to go in and out, waste products to go in and out, hormones to go in and out, etc., etc., etc. So there's a big surface area of coverage for the blood. So it's almost more or less an ideal system. It has big transport routes like arteries and veins. You have big transport routes like the motorways, which split into smaller roads which reach more parts of the country, eventually down to little tiny streets which reach all the different houses in the country. So this is exactly how the system works as a transport system. Okay, I'm going to talk about the arterioles now. We've had a look at the arteries and the veins. Let's talk about these things called the arterioles. Now they're small, they're microscopic. They're 10 micrometers to 50 micrometers. That's 10,000 to 50,000th of a millimeter. Okay, so that is small. They're made up of smooth muscle and some cells, and they have very, very important roles. First of all, they're the vessels of precapillary resistance. That means they provide a resistance to blood flow before the blood gets into the capillary system. So they can moderate the blood flow and the blood pressure before the blood gets into the capillary, because obviously you don't want high blood pressure going through a delicate capillary network. Also, you want to be able to control how much blood goes through a specific tissue at any one time. Because, say for instance, you only need your capillaries in your digestive system to have a good supply of blood when you've eaten a meal. You don't need it when you haven't got food in your system. So arterioles will provide this control. And the other thing that they do is that they smooth out the pulse flow of blood and make it a continuous flow. So if I show you along the bottom here, as blood leaves the heart, it pulses because of the pulse of the heart. As it gets down towards the arterioles, and they start to branch out, then it eventually gets to the arterioles, and they reduce that pulse to a nice steady flow. Because if it was a pulse flow going through your capillaries, they wouldn't be as efficient as exchanging things, because they need a nice steady flow. The next diagram shows you an arteriole and a venule joined together by the capillary circulation. Now, the thing I want you to notice here is that the capillaries have a number of different ways in which they can control blood flow. So for instance, if you look here, these are knots of muscle. These are what we call precapillary sphincters. So these can close and divert blood to different parts of the tissue. So if you wanted to reduce the amount of blood going into the tissue, you could actually close these off. However, when you close them off, the blood can flow directly through what are called shunts. And this is an AV shunt from the artery to the venule, arteriole to the venule, and this AV shunt means that blood flows directly through. It doesn't take a long route through the tissue. Now that picture I showed you right at the beginning of the vascular system, and I'm going to go back to it now, is quite important. Here. Because what this shows you, and it shows you it really, really neatly, and this is why it's one of my favourite pictures, is, look here, 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 and there's another one. There's plenty of them here, well, there's loads here. Those are the sphincters we talked about. Those are the capillary sphincters. So those are parts of this which can stop the blood going into the capillary network. And you can often see at the base here how pinched, this one, how pinched that can be. So, and there's another one here. Okay, so it's a really, really neat system. And this is controlled via our dear friends, the sympathetic and parasympathetic nervous system. They allow for blood to pass through the capillary beds and control blood passing through the capillary beds, as well as a number of different chemical factors that also influence that blood flow. The next slide is equally amazing, I think, because this shows you how complex capillaries can be. So here are blood vessels. So these are the big blood vessels, arteries and veins. And over here, the smaller blood vessels, arterioles and venules. And then here, this huge, huge mass of capillaries, this huge network of capillaries. This means that this whole area is literally bathed in one big sheet of blood going through this capillary network. And capillaries are very, very important. There are different types, and I've got them here named. You don't really need to know. It's just the fact that some of them have gaps in their membrane so that blood can pass through in and out easier. Some of them don't have complete membranes and provide just big spaces full of blood called sinusoids, and you find that in your liver. And others are continuous, so the blood flows simply through a vessel just like it would through any other vessel. But they're small, 6 to 10 thousandths of a millimeter. So all that blood that's pumped out of the heart gets disseminated and distributed via these capillary networks. Okay, these capillary networks which cover our bodies. Okay, for the final part of this session, we've looked at the heart, we've looked at the blood vessels. We need to have a look at an important clinical parameter which is determined by those two things, and that is our blood pressure. Okay, so this diagram shows you all the determinants of arterial blood pressure. Okay, these are all the things that will affect arterial pressure. Okay, it looks complicated. It is complicated, but hopefully I can simplify it by doing this. There are 3 important determinants of blood pressure. Cardiac output. We've already seen that. You know what cardiac output is. That is how much blood is pumped out of your heart over a time period, such as a minute. So that's the first thing that determines your blood pressure. How much blood is being pumped around the body. Now the second thing that determines blood pressure is something called peripheral resistance. Okay. The best way to explain peripheral resistance is for you to think about what happens when you pass water through a hose, through your garden hose at home. You turn on the tap and the water flows through the hose. The pressure of the hose, the pressure of the water coming out of the hose is the same unless you do something like stand on it or squeeze it. And if you squeeze your garden hose, what do? You increase the resistance to the water. And if you increase the resistance to the water, one of the things that happens, and if you squeeze the end of your hose, you know that, is the pressure of the water gets higher. So similarly, blood passing through your blood vessels, the pressure becomes higher if the resistance to that blood is higher, if the vessels are squeezed, if the vessels are thinner than they normally are. So that's the second factor. The third factor is blood volume. Okay. This is a determining factor because, again, if you think about blood passing through blood vessels, okay, the more blood there is in the system, the higher the pressure. So this is analogous to you turning your tap higher and higher and higher on your garden hose. So with the same hose, if you turn the tap on a little bit, there's a smaller amount of water passing through the hose. It's at a lower pressure. When you increase the amount of water going through that hose, that increases the volume, and that increases the that increases the pressure. So, 3 things determine your blood pressure: your cardiac output, how fast your heart is beating, how much blood is beating at one time; the peripheral resistance, how open or closed your vessels are, particularly your arteries and arterioles; and the blood volume, how much blood there is in the system. Any of these change, then you can change blood pressure. So an increased cardiac output will increase blood pressure, increased peripheral resistance will increase blood pressure, and increased blood volume will increase blood pressure. Decreased cardiac output can decrease blood pressure. Decreased peripheral resistance can decrease blood pressure, and decreased blood volume. Okay? And that's the way in which the body can control how much blood is traveling at what pressure around the body, by those 3 things. So how does it do it? How does it control the blood pressure? First of all, there's a strong central nervous system control of blood pressure. Basically, there are a number of things that you can control your blood pressure through. There's a number of reflexes, homeostatic mechanisms that can control blood pressure. Now, pressure can be sensed, and we saw it in one of the very first lectures, in things called baroreceptors, in receptors that respond to pressure. We can also respond to chemical changes in the blood, and that will change blood pressure. And we can also influence our blood pressure by inputs from higher brain centers, like emotions. So stress, for instance, can increase our blood pressure. Now, all of these things work by sending nerve impulses along spinal nerves T1 to L2. Okay, remember that? Thoracic to lumbar regions. So all of those spinal nerves have nerve fibers that go out and attach to blood vessels. What they do is they basically attach to the smooth muscle of the blood vessels. So if they stimulate it, it contracts. If they don't stimulate it, it relaxes. So the nervous system can control the amount of resistance in your blood vessels by stimulating smooth muscle. Okay, so this is a more detailed picture of what we saw in that feedback loop we talked about in terms of blood pressure. So these are receptors. Okay, so these are the receptors in the aortic arch and the carotid sinus, and they basically feed information to the brain about blood pressure. So we get an increased pressure sensed in here. So if there's an increased pressure sensed in these 2 areas, then the receptor basically sends a message to the brain to inhibit vasomotor center and to inhibit cardiac center. That reduces the heart rate, and it also opens up or dilates the blood vessels. So the nervous system slows down the heart and opens up the blood vessels. And that means you get a reduction in blood pressure. Now that happens often when you stand up. So if you stand up, you get postural changes in blood vessel pressure. Here, this is a decreased pressure. So for instance, when you stand up, blood drains away by gravity towards your feet. But as the baroreceptors, what the baroreceptors do here is they detect that decreased pressure. Get rid of that. Decreased pressure. Baroreceptor that excites constriction increase. Okay, so all I've done is just changed it to a decreased pressure. The baroreceptor here, the baroreceptors here, here, detect that drop pressure, they excite the cardiac center, they excite the vasomotor center, which causes an increase in heart rate and a constriction of the blood vessels, therefore increasing the blood pressure. This ensures that there's enough pressure to pump the blood around the body to overcome the gravitational drop of blood due to standing up. So it's a very, very elegant way of showing you the homeostatic feedback loop, how the brain and the nervous system control blood pressure. Okay, there are some other influences on blood pressure, and I'm going to show you them here. So for instance, blood pressure can be changed according to the level of carbon dioxide in the body. And there are special receptors in the bloodstream and in the central nervous system that measure the chemical composition of blood. They're called chemoreceptors. They're effectively measuring how much carbon dioxide is present. And if there's too much carbon dioxide present in a tissue, that would suggest there's not enough blood going through there. So to respond, the body would increase blood pressure. Now the other thing you know is that, and you know this yourself, that emotions can control blood pressure. And that's because higher brain centers can influence blood pressure. And that's all part of the body's stress response. It's fright, flight, and fight response. So they can have overriding effects on blood pressure. Okay, let's go back a few slides and have a look. Remember this diagram. Remember we said, and now we know, the mechanisms of cardiac output, the control cardiac output. We know about peripheral resistance now, so we know that actually that the nervous system and hormones, etc., can control the constriction of blood vessels and therefore change pressure. The final bit we need to know about is blood volume. Now the key determinant of blood volume is obviously the amount of fluid in the circulation. So the final thing that we need to talk about is the kidney and blood volume changes, because it's through the kidney that we can change blood volume, and through a number of hormones, antidiuretic hormone, angiotensin, and atrial natriuretic peptide. You don't need to know about those at this moment in time, but those three hormones all act to regulate the amount of fluid in our blood. So it works rather like this. If we need to increase our blood volume, then we can retain fluid in our blood. If we need to decrease our blood volume, we can release fluid from our blood in our urine and therefore reduce the blood volume. And by reducing blood volume, we reduce blood pressure. Okay. Now just to show you that this is all relevant, this is a slide that shows you, and it comes from the British National Formulary website, explaining about how you should prescribe blood pressure medication. What it's showing you is the different types of drugs used to control blood pressure. So we have ACE inhibitors or angiotensin receptor blockers. Now that's effectively a blood pressure medication that affects the size of blood vessels. Yeah. Beta blockers, they affect the excitability of the nervous system. Calcium channel blockers, they affect the excitability of the heart and the vascular system. And diuretics, they make you go to the toilet more often and reduce blood volume. So a comprehensive blood pressure medication seen here in resistant hypertension is to give a combination of all of these. Why? Because what you're doing is you're influencing all of the different components of blood pressure. You're influencing cardiac output, peripheral resistance, and blood volume using these drugs. So if you need to know more about this, go to the BNF and find out about what these drugs are and what they do. But take it from me, they are working by mimicking the physiology of the cardiovascular system, by controlling blood pressure by one or more of the three key determinants: heart rate, stroke volume, peripheral resistance, and blood volume. Okay, so that's the end of our session today, and I'll be back for one more next week, which is the digestive system.